Hi, my name is Jonathan. I am a student at the Autonomous University of the State of Morelos, and I'm going to present the project entitled Metabolic Regulation of CD4 T cells Activation and Function, revealed by computational modeling. Well, in this project we use logical modeling to study the cross-regulation of metabolisms and the activation of CD4 T cells. In the model, the components of a biological system are represented by nodes and links, and the nodes are associated with a variable that represents their activation. Once the model is validated, we use it to perform analyses like the computation of stable states and perturbation analysis representing knockouts and knock-ins scenarios. Our model was generated and validated for the adult CD4 T cell activation scenario, and then it was used to explore the neonatal CD4 T cell scenario. And we predict that the metabolic state of neonatal CD4 T cell can affect their activation and function. To make our model, we first review the bibliography or databases looking for information related with our project. Later, with the help of computational tools like GenSim, we can integrate this information in a model and we can complement the model with experimental data. After with the model, we can do dynamic analysis like the stable states, and if these states match with the bibliographic information, we can make prediction about the biological system, but if this state doesn't match, we go back to the bibliography and repeat the process. This is our current model, which is composed of 212 nodes, and the nodes representing genes overexpressed in neonates are shown in sky blue. Our model can be divided in two parts. One of these contains the activation pathways and the other the metabolic pathways. And the model contains complete metabolic pathways like the glycolysis and the tricarboxylic acid cycle. The model contains pathways that connect the activation and the metabolisms, like the PI3K AKT mTOR pathway that is activated by TCR and CD28 signaling. Now, when we computed the stable state of the wild-type model, we get 6 stable states. The first two states, E1 and E2, represent the phenotype of naive T cells with the TCR signal off, and these cells doesn't have an increase in the metabolic rate. The third state, E3, represents the phenotype of an energetic T cell because this state has activated the TCR signaling but doesn't have the co-stimulation of the CD28. In the last three states, the TCR and CD28 signaling are activated. So these cells are effector T cells and secrete pro-inflammatory and anti-inflammatory cytokines. But in the state E4, the metabolic reprogramming doesn't occur, so the cell is not activated correctly. And in the E5 and E6 states, the metabolic rate increases, so these cells have an anabolic program. Next, we compute the stable states when lactate has a permanent high level, and we obtain 5 states. The first three states, E1, E2, and E3, are very similar to the first three states obtained with the wild-type model, except obviously for the lactate, which have a value of 3. In states E4 and E5, high lactate levels cause a decrease in glycolysis, inhibiting the biosynthesis and cause a decrease in ATP synthesized by oxidative phosphorylation. Also, the cells can produce pro-inflammatory cytokines, causing an anti-inflammatory response. And the deficient metabolic phenotype, represented by the E4 and E5 states, have been represented in lactate-rich media, such as the extracellular medium of cancer cells. And to end, our results show that exists a cross-relation between metabolic and T-cell activation pathways. And our computational model was useful to demonstrate the impact of metabolic pathways on T-cell activation and function, and important role in neonatal CD4 T-cell activation. And now we will use the logical model to find strategies to improve the immune response of neonatal CD4 T cells.